Darwin's book is On the Origin of Species, and to quote from David Galanter's essay, quote, "There's no reason to doubt that Darwin successfully explained the small adjustments by which an organism adapts to local circumstances, changes to fur density, or wing style, or beak shape. Yet there are many reasons to doubt whether he can explain the big picture, not the fine-tuning of existing species, but the emergence of new ones," close quote. Okay.
Evolutionary developmental biology is the subdiscipline concerned with how changes in embryonic development during single generations relate to the evolutionary changes that occur between generations and lineages. Noticeably, creationists neglect non-animals when discussing macroevolution, or at least their concept of what macroevolution is. So as a segue into this topic, let's talk about plants. We all know that the different fruits and vegetables that we grow or buy at the market are derived from different parts of their respective plants. And some of us even know that we have subsequently altered, accentuated, and exaggerated the yield and nutritional capacity of these plant organs from their humble wild counterparts. Perhaps the most well-known example of directed plant evolution is that of the wild mustard plant, Brassica oleracea, and the many vegetable crops derived from its anatomy. B. oleracea varieties include important vegetable crops, such as cauliflower, cabbage, broccoli, kale, Brussels sprouts, collard greens, savoy, kohlrabi, and gailan. These varieties display very high phenotypic diversity, even though they originated from the same ancestor. Cauliflower and broccoli exhibit extremely enlarged inflorescence meristems and or flower buds, which form a major edible organ called the curd. Cabbage and Brussels sprouts display extremely enlarged terminal leaf buds and lateral leaf buds, respectively. The leaves of kale and collard greens are obviously different from those of uncultivated wild cabbage, whereas kohlrabi has a dramatically enlarged stem, which is its edible organ. The completion of the B. oleracea genome sequence and assembly revealed that it and other Brassica species exhibited a whole genome triplication with respect to the genome of the related species commonly used in plant research, Arabidopsis thaliana. Except for the specialized organ, the curd, the phenotypes of cauliflower are similar to those of closely related species in the genus Brassica. Consequently, identifying genetic variation between cauliflower and closely related species is important for uncovering the phenotypic specificity of cauliflower. Therefore, after applying the methodology of comparative genomics to cauliflower, it was observed that 2,116 gene families were cauliflower-specific, while only 6 and 2 gene families were significantly expanded and contracted, respectively. Importantly, 1,065 positively selected genes were identified in cauliflower. Of these, novel genes involved in auxin biosynthesis might have been generated during the evolution of cauliflower, while the genes involved in cytokinin biosynthesis were contracted. Auxin and cytokinin are two important phytohormones that mainly function in regulating plant growth and development. The adjustment of the homeostasis of auxin and cytokine in phytohormones in cauliflower relative to that in other B. oleracea species might be associated with the phenotypic characteristics of cauliflower, since curds are floral organs and since vegetative and reproductive growth phases oppose one another. Ironically, the types that reject evolution tend to also misuse the biology of hormones to make claims about fundamental differences between things. Thus, there exists a link between genetic alterations in development via the function of signaling pathways already present, rather than the input of new information, and the evolution of new traits in populations. Evo-devo. The same processes at play in the evolution of the genome, and consequently, the form and phenotype of plants, are involved in the evolution of all organisms' diversity. Mutations happen naturally in domesticated plants and animals through physical and biochemical processes. Then humans select the individuals that exhibit their associated phenotypes for breeding and propagation as variants in the gene pool. The mechanism is no different in nature. The ability to catch prey animals or outrun predators can also select variations in a population just as readily as a domestic breeder or farmer. The mutations that predispose racing whippets to high speed are as naturally occurring as the ones that enabled wild pronghorn, as they diverged away from their close ungulate cousins, giraffes and camels. In the former case, humans found those mutations. In the latter, predators did. Even bacteria, that are somewhat artifactually and erroneously thought of as single-celled organisms, because of the way in which those most familiar to us are isolated and cultured, are more commonly found in multicellular biofilms consisting of one or more species in nature. Could this be the foundation for the evolution of true multicellularity in plants, animals, and fungi? Of relevance to the topic of evo-devo and animal macroevolution are Hox genes. Ed Lewis studied fruit flies for decades. His hypothesis about the relationship between the serially homologous segmentation of insect bodies and possible underlying gene duplication was a specific deduction from general evolutionary theory. More primitive winged insects which are seen earlier in the fossil record don't exhibit quite as distinct metamorphic stages in their life cycles. Perhaps not coincidentally, such insects, like dragonflies or grasshoppers, also have two wing pairs. While insects with distinct metamorphic stages have one pair of wings and a pair of accessory structures. However, rather than being the result of entirely new genes and new information for new structures, both the development and evolution of modified segments and accessory appendages instead stems from modifying extant genes, pathways, and structures. Creationists notoriously misunderstand and/or misrepresent these classic genetic experiments. The goal was never to evolve some new animal from fruit flies via mutation, as they claim. Instead, given what was already known about entomological diversity, it was observed that certain Hox knockout mutations reversed the fly's wing morphology to a more primitive forewing layout. These experiments were working backward, deconstructing a phenomenon to understand its mechanisms, rather than trying to fast-forward evolution in a laboratory. Such genetic biochemical dissections follow the same logic that was used to figure out metabolic pathways and the genetic code in the mid-20th century. In any case, the ancestral Hox gene, singular, was first duplicated in the common ancestor of bilaterians. Subsequent duplication events produced multiple paralogs, and then later on, in vertebrates, this array was duplicated multiple times as whole gene clusters. Don't forget that even we humans are segmented. This is the paradigm for how a basic modular domain is expanded through evolution and combined with other modular domains to form a functional protein, followed by families of related proteins that are expanded further still en masse to yield protein superfamilies. That perform different but related functions in different but related organisms. As another example, teleost fish underwent an additional whole genome duplication early in their evolution. Teleost fish have 7 to 8 Hox clusters. Perhaps not coincidentally, half of all vertebrate species are teleost fish, and teleost fish are the most morphologically diverse group of vertebrates. Finally, a lineage within teleosts, the family Salmonidae, has an additional duplication resulting in 13 Hox clusters within that clade. Compared to only 4 Hox clusters present in the human genome. Evo-devo at least partially explains the apparent disparity between small-scale microevolution and macroevolution. Large-scale genomic macromutations like gene, as well as both partial and whole genome multiplications are involved. Alterations in aspects of normal development, gene expression and physiology that mediate developmental changes are the same sort that underlie morphological or macroevolutionary change. Thank you for watching.